Okay, students, as you all know, there are different types of organic molecules in the living matter, and those organic molecules are known as biomolecules. There are different types of 4 main types of biomolecules present in living organisms, and the second type is protein. That is what we are going to discuss now. So, as you can see, protein is an essential constituent in all living cells. It is present in all the living cells and it mainly makes up different parts of the living cells. So, that is why we say it is an essential constituent. And if you look at a mature human body, there is about 17% of protein. So, 17% of the mature human body is composed of proteins. And out of the 4 main elements, all 4 elements are present in protein. So, the constituent elements of protein is carbon, hydrogen, oxygen and nitrogen. So, it is made up of C, H, O and N. And also sometimes there can be sulfur. So, the main components are carbon, hydrogen, oxygen and nitrogen. Sometimes there will be sulfur. So, these 4 elements combine and form a molecule or compound called the amino acids. And these amino acids combine in large number and form the protein molecule. So, protein is a complex molecule because it is a very large molecule and they are polymers. So, as I said, the small units which are called the amino acids, these are the basic structural unit of proteins. They are called amino acids. So, a very large number of amino acid molecules combine together and form the protein molecule. Therefore, it becomes a very complex structure, a complex molecule. And based on that, there are different properties for protein molecules. And if we look at different types of food that are rich in proteins, we can consider meat, fish, egg white and some cereals. These are all rich in protein. Now we will see how these amino acids are formed and the types of amino acids present. So, if you look at this structure, this is given in your book, amino acid structure will look like this. In this, there is a carbon in the middle. The NH2 group, this group is called the amine or amino group. And the group COOH is known as the carboxyl group. This comes from the carboxylic acid type of acids. Therefore, this whole molecule is known as amino acid. And in addition to that, always there is a hydrogen in amino acid. So, these 3 groups are fixed. The NH2 group, hydrogen group and the COOH group are always there in an amino acid. But this last group, which is denoted by R, it is known as an alkyl group. Here you can see R represents a group containing both carbon and hydrogen. And we call that as an alkyl group. Now this group is different from amino acid to amino acid. Due to the change in the R group, there are 20 different amino acids which are present in living organisms. So all proteins present in organisms from bacteria to human are made up of these 20 different amino acids combined in different sequences. So when they combine in different number and different order, that is what we refer to as sequence of amino acid. That will give rise to different types of proteins, and they will have different properties and different uses. So here the simplest amino acid is called glycine, and the R group in the glycine molecule is a hydrogen atom. So this is glycine, which is the simplest amino acid present. Some amino acids cannot be synthesized within the body. So inside the human body, some of the amino acids cannot be synthesized. Therefore, they have to be taken from outside. That is, they have to be consumed in the form of food. So, any food that is rich in proteins will contain these amino acids. And the amino acids that have to be consumed in the form of food are known as essential amino acid. So students, now you have a good understanding about proteins. They are made up of carbon, hydrogen, oxygen and nitrogen combined to form amino acids. Different amino acids combine in large number in different sequences to produce different types of proteins. So now we will look at the different types of proteins present in living organisms. So here the different proteins present in both plants and animals. If we take the first example, in muscles. In human muscles, you all know muscles are the tissues that are present in our hands, legs, the mouth. So to do some activity, to make a movement, to talk, to move your hand, to move your leg, you need to move your muscles. So these muscles have to stretch and contract. So that is possible because of these two proteins, actin and myosin. These are the proteins present in muscles and they help to give flexibility to muscles. Then the next type of protein is present in bones. In bones, there is osine. Osine is a protein that gives the structure and the strength to the bones, which provide a structure for the body. With the presence of bones only, we have a certain shape and we are able to move our limbs. The third type of protein is present in red blood cells. Already we looked at this when we discussed the different types of elements present in a human body. We said that there is iron in hemoglobin, and hemoglobin is an important component of blood that transports oxygen throughout the body. So when we say hemoglobin, heme refers to the iron and globin is the protein. So red blood cell has hemoglobin, in that globin is the protein. The next one is the protein present in hairs. As you know, mammals have hair on the skin. There are hair on your head, the hair all around the body. This is there for protection. Protection of the skin from cold weather and other protection. So this protection is given by the keratin protein. One of the components that help to give the protection is the keratin protein that is present in hair. So these are all presence of protein in animals. Now here you can see two examples where protein is present in plants. In leguminous food, when we say leguminous food, beans, long beans, soya beans, all these are known as legume plants. So these legumes contain a protein called legumin. So the name itself refers to the protein. Legumin is the protein that is present in legume plant products. That is beans, long beans, soya beans. So that provides us with lot of protein. When we consume this food, we get lot of protein. Then there is protein in wheat. Wheat has gluten protein. That is another type of protein that is present in wheat. And finally, we said the protein-rich food. One of the protein-rich food is egg white. And this egg white contains a special type of protein that is called albumin. You have this experience of frying egg. Initially, when you break the egg white looks colorless. It looks transparent. But when you fry the egg, this becomes white in color. That is why we call it egg white. And that is due to the albumin protein present in it. There is a change taking place to the protein present in egg white when we fry it. Okay, students. Now, these are the examples of different types of proteins present in plants and animals. Now, we will discuss the significance of proteins as an energy source. As we have discussed before, carbohydrate is one of the main energy sources present in living organisms. In addition to carbohydrates, lipids also act as energy sources. But at an instance where there is not enough carbohydrates or lipids in the body of a living organism, then the protein will act as an energy source. Only when there is insufficient carbohydrates or lipids, protein will act as the energy source. So, if I note it down, when there is insufficient carbohydrates or lipids, then the protein will act as the energy source. Otherwise, normally the body of living organisms does not utilize protein as an energy source. To make structural components. When we discuss cellulose, we looked at the structure of a cell. In both plant and animal cells, there is an outer boundary which is a membrane, and that membrane is called the cell membrane. One of the main constituents of the cell membrane is protein. So, protein is a constituent of cell membrane. Of the cell membrane. In addition to that, different types of proteins are present in different parts of the living organism's body. For example, if we take the hair or feathers of birds, then there is a protein called keratin in them that provides protection. So, keratin is a protein present in hair of mammals and feathers of birds. Therefore, protein is a structural component. So, here another example, keratin. It is present in hair and feather of birds. So, there are two types of proteins. One type as a constituent in the cell membrane, the other type keratin that is present in hair as well as feather of birds. As enzymes. Now, this is a special type of protein that is produced within the body, and the function of this protein is to increase the rate of biochemical reactions taking place inside the living body. After this, we will be discussing the property and function of enzymes. So, enzymes are special types of proteins. So, these are special proteins that increase the rate of biochemical reactions. And remember students, all enzymes are made up of proteins. As hormones. Hormones are chemical substances that involve in homeostasis and coordination. These are two new terms for you. As you know students, birds and mammals are warm-blooded animals. Warm-blooded means our body temperature does not change with the change in environment temperature. So that means the temperature of the human body as well as other mammals and birds are maintained at a constant value. So this process of maintaining certain factors of the body is known as homeostasis. And all the processes that take place in order for the body to function properly is known as coordination. And these substances that involve and carry out homeostasis and coordination are known as hormones. There are some hormones that are made up of proteins. So, as hormones, these help in homeostasis and coordination. But remember students, there are some hormones that are protein hormones and there are some hormones that are lipid hormones. We will discuss that when we discuss lipids. So again, to summarize, all enzymes are proteins but only some hormones are proteins. Those two are two different significance of proteins. Then protein acts as antibodies. As you know, our body always protects us from external factors. So the pathogens that enter our body are external factors that can harm our body. In order to prevent that harm, our body produces a chemical substance called the antibodies. This is done by the white blood cells present in blood. So these antibodies are made up of proteins. So here antibodies are produced against pathogens. So this is a process where our body fights against pathogens. So now students, you have seen all the significance of protein as an energy source to make structural components, as enzymes, as hormones, and as antibodies. Now that you have understood the structure and the significance of proteins, we will discuss how we can identify the presence of proteins in different food samples. So this test for proteins or test to identify proteins is given a special name. It is called the Burette test. And for this test, we need these materials. The first one is a food that contains protein, a food rich in proteins. So for this, we can use any type of cereal or lentil, dal, mung atta, green grams or any type of lentil or the egg white. So these are all food rich in protein. So here we will be using dal or any other type of food that contains protein. And to grind this food and extract the protein solution, we will be needing a mortar and a pestle. Mortar and pestle for the purpose of grinding the food item. And then we need two reagents or two chemical substances to do this activity. One is sodium hydroxide solution. You can directly use the sodium hydroxide that is available in the lab, or you can dissolve sodium hydroxide pellets in water and prepare a solution of sodium hydroxide. Similarly, we need a solution of copper sulfate. Here you can see the blue color is hydrated copper sulfate. We can dissolve this in water and get the copper sulfate solution. So we need copper sulfate solution. And to carry out the test, we need a test tube. The method in which we are going to carry out this test, we need to first get the solution of protein. So for that, we take a small amount of, let's say, dal into the mortar. We add a little bit of water, a very small amount of water, just to grind it and extract the protein from the food item. And then you have to nicely grind the dal with water using the pestle. In order to facilitate the grinding, we can even soak the lentils in water for 2 or 3 hours or even overnight. But if we are using the egg, so it's just a matter of breaking the egg and taking out the egg white separately into a test tube. So once either we can take the egg white into the test tube, or like I said, if we use dal, we have to grind it well and then we transfer the solution into the test tube. And we keep that aside. Also, we need to prepare sodium hydroxide solution and copper sulfate solution. Then what we do is we take equal sizes of test tubes. The normally test tubes are of equal size. Into one tube, we already have the protein solution, and in the next tube, we need to take equal volume or excess of sodium hydroxide. It has to be equal or more than the protein solution. Then we transfer the sodium hydroxide solution into the protein sample. Thereafter, we add a few drops of copper sulfate solution. Then you will get the required observation if protein is present. So, I will summarize the procedure again. So, in the method, first we will have to make an extract of protein, or we have to extract the protein from the food. So, first a solution of protein is extracted. This is done by grind the food with a small amount of water using the mortar and pestle. So, we have the protein solution. We take it into a test tube. Take the solution into a test tube. So now we have the solution that contains protein in a test tube. Then what do? We have to add equal or excess amount of sodium hydroxide. Add equal or excess amount of sodium hydroxide solution. You have to remember, students, either the volumes have to be equal or sodium hydroxide has to be slightly in excess. You can't add less sodium hydroxide. So, it has to be either equal or excess volume of sodium hydroxide. Then to that mixture, we add a small amount or few drops of copper sulfate. So, to that solution, add a small amount of copper sulfate. Copper sulfate solution. Then you will get the required observation. So, students, this is a very simple activity. where we test the presence of protein in food sample. Now that you have understood the steps clearly, let us go and carry out this activity in the laboratory. Okay students, now the activity to test for the presence of protein in food samples. As you know, there are different types of food that contain protein. Dal is one of those substances. You can also use egg white for this activity, but for now I'll be using dal. Since dal is a solid substance, we will have to get an extract of the dal. For that, I will need a mortar and a pestle. First, I am going to transfer a small amount of dal into the mortar and pestle. And I will be adding a little amount of water to it. I will have to grind it well and get the extract. To make it easier for grinding, you can also soak the dal in water for about 2 to 3 hours. Then it will be more softer and it will be easier to grind. So now I am going to grind it and get the extract. You will have to keep on grinding it for quite some time so that the substance from dal gets extracted into the solution. Now I have ground the dal into a nice solution and extracted the substance from it. I will decant this solution. Decant means just transfer the solution into a beaker. Now I will not be needing the mortar and pestle. I will put it aside. Now we are going to do the test to identify the proteins present in the dal solution. For that, I'll be needing 2 reagents. You all know the reagents. This is copper sulfate. Copper sulfate is blue in color. It is a nice blue color solution. And sodium hydroxide. First, we will need the sodium hydroxide solution. I'll be using a test tube, taking a small amount of the dal extract and into another test tube, you will have to take either equal amount of sodium hydroxide or slightly excess amount of sodium hydroxide. So I'll just use the 2 test tubes to compare. Here you can see I have taken slightly more sodium hydroxide than the dal extract. Now I will transfer the sodium hydroxide into the dal extract. You can still see the color of the dal extract. Now to this mixture, I will be adding a few drops of the blue color copper sulfate solution. Now observe clearly what will happen. You all can see a nice color change, the appearance of a purple color. So with sodium hydroxide and copper sulfate, if a purple color appears, that means that sample contains protein. And you know, students, what is the name given for this test? Yes, this test is called the Burette test. Okay, students, now you observed the activity clearly and you got the observation, you saw the color change very clearly. So, if you do this activity, that is the Burette test, to test the presence of protein in food, what is the observation that you should get? As you add the blue color copper sulfate solution, the solution has to turn purple. So the observation will be appearance of purple color. This will be the observation of the activity. As you add the copper sulfate, the purple color will appear. So this you can use to conclude that there was protein in the sample. That means there is protein in the lentil that you took, that is dal, or any other food item that you can use to test will contain protein if you obtain the color purple. If appearance of purple color confirms the presence of protein. So the conclusion will be there is protein in the sample. Okay students, so now you have got a good understanding about what proteins are and how to identify the proteins in food. Now when we discuss the significance of protein, we said that protein is present as enzymes. So that is what we are going to discuss next. So enzymes. The special proteins, they are called organic catalysts that are produced within the organism to increase the rate of biochemical reactions are known as enzymes. So we will look at the different terms that are important to us from this explanation. First, if we look at what biochemical reactions are, I told you all once before, any chemical reaction that is taking place within the body of a living organism is called a biochemical reaction. Because the living organism is a biological system and a chemical reaction is taking place inside, so it is called biochemical reaction. And these biochemical reactions occur at a certain rate, at a certain speed, and also they have to take place within the body. For example, if we take man, the normal body temperature of a man is 37 degrees Celsius. So these biochemical reactions have to take place in that temperature. So therefore, we need a special substance. This is an organic substance and that is called a catalyst. So the substance that increases the rate of reactions is called a catalyst. So here it is an organic compound that is a catalyst and it is given the special name enzyme. And all the enzymes are made up of proteins. This is important. All enzymes are made up of proteins. They are made up of proteins. They increase the rate of chemical reactions and also it is the biochemical reactions. So therefore, we can call these enzymes as biological catalysts. The enzymes are known as biological catalysts. So the activity of the enzyme is to catalyze biochemical reaction. As an example, if we look at a reaction, as you saw in the test for sucrose, to convert or break down sucrose to form glucose and fructose, that is the hydrolysis of sucrose, we had to heat it with sulfuric acid. So when we say heat, we are providing a high temperature. But the same reaction when you eat food that contain sucrose. So example, you eat a little bit of sugar. This sugar contains sucrose that has to digest within your body and it will be converted to glucose and fructose. This process, the process of digestion, is a biochemical process and that is catalyzed by an enzyme. So here as an example, we can say the hydrolysis of sucrose, if done in the lab, we need to heat with sulfuric acid. But in the digestive system, it is catalyzed by an enzyme at a less temperature. So as you can see, the catalyst, that is the enzyme, helps to carry out the reaction at a lower temperature and at a faster rate. Next, we will see how we can demonstrate the activity of an enzyme. So, to demonstrate the activity of enzymes, we use the enzyme amylase and we demonstrate the activity of amylase on starch. In a name, if you get these three letters at the end as A's, amylase, sucrase, invertase, they all mean that it is an enzyme. And the first part of the name usually refers to the substrate they act on. Here, amylase is for starch and enzymes are usually specific to a certain substrate. One enzyme for one substrate or more than one enzymes for a certain substrate. So, they are very specific. The action of enzyme or activity of enzyme is very, very specific. And here we use these materials to demonstrate the activity of amylase on starch. So, as a starch containing food, we use wheat flour. So, here we are going to use wheat flour as the starch containing food. Wheat flour. Then we use iodine solution. Remember students, we used iodine solution before. When we add few drops of iodine solution to a substance containing starch, there was a color change. The reddish brown color of iodine becomes dark blue or dark purple in color. That confirms the presence of starch. So, here again we will be using iodine solution. This activity has to be timed. We need to repeat a certain procedure within 2 minutes time interval. So, to do that, we use a stopwatch. Then we use a test tube to carry out the reaction. And this is a white porcelain tile. This tile is used to take a sample and to observe color change clearly. A white porcelain tile. And finally, we use germinating mung seeds. Germinating Now you will be wondering, we said the activity of amylase, and we don't have amylase as a material. Yes, you all are correct. That is why we are using germinating mung seeds. Now if you remember, students, in germinating seeds we said there is maltose. But as you all already know, the green grams contain starchy substances. And when they start, the seed contains starch, and when it starts to germinate, as it starts to grow, there is amylase enzyme acting on the starch and converting it to maltose. That is why the germinating seeds normally contain maltose. And for this, there is amylase enzyme in the germinating seed. So what we will be doing is, we take the germinating mung seeds into a mortar and a pestle. That is not shown here, but you are familiar with those two equipments, the mortar and the pestle. We take mung seeds into it, add a little bit of water, grind it very well and extract the amylase enzyme. So then we filter the solution and take it as the amylase solution. We will be using that for the activity as the enzyme solution. At the same time, we take a small amount of the wheat solution, that is wheat flour, and we dissolve it in water. But for this activity, since we are going to repeat it for a period of 20 minutes, we will need a certain amount of wheat flour solution and germinating mung seed solutions. So we take them in equal volumes into 2 test tubes, and just before mixing the 2 solution, we should be ready to start the stopwatch. So as we mix the germinating mung seeds, that is amylase solution, with the wheat flour solution, that is starch solution, we start the stopwatch. So we mix it and keep it aside, and we observe until the time becomes 2 minutes. And at the end of 2 minutes, we take a drop out of the reaction mixture, put it on the white tile, and on that we put a drop of iodine. You will observe that the reddish brown color of iodine will turn into a dark blue black color or dark purple color. That indicates the presence of starch. So that means after first 2 minutes, when you drew out a sample from the mixture, there is starch present. Then we wait for another 2 minutes and repeat the procedure. Take out a drop from the reaction mixture, put it on the white tile, add a drop of iodine. Again you will get a dark blue or purple color appearing. But you can see, when we keep on repeating this step every 2 minutes, the dark blue or purple color will decrease in intensity. It will become lighter and lighter. And we keep on repeating this procedure for 20 minutes. And after 20 minutes, when we take a drop from the reaction mixture and test with iodine solution, you will not see a blue color forming. Okay, I will summarize these steps again. So the first step, prepare a solution of amylase. That is grind germinating mung seeds and filter the extract. So by doing this, we can prepare a solution of amylase. Or if you have a solution of amylase in the lab, we can use that directly. Then we need to prepare a solution of starch. So the second step, prepare a solution of starch. What do we do after that? We need to take 2 milliliters of the starch solution into a test tube. Take 2 milliliters of the starch solution into a test tube. To that, we have to add 2 milliliters of the amylase solution. Add 2 milliliters of amylase solution to starch. And while we do that, we need to start the stopwatch. And start the stopwatch simultaneously. So that you can time the activity. Then we need to wait for 2 minutes. We draw out a drop of the reaction mixture, place it on a white porcelain tile, and to that we need to add a drop of iodine solution. So after 2 minutes, draw a small amount of the reaction mixture and place it on a white porcelain tile. To that, what do? We add a drop of iodine solution. Add a drop of iodine solution. Then what do? We wait for another 2 minutes and repeat the same steps. Draw 1 drop of the reaction mixture, place it on a porcelain tile. To that we add a drop of iodine solution. Like this, we need to repeat the steps every 2 minutes for 20 minutes. So the last step, repeat the steps every 2 minutes for 20 minutes. Repeat the steps every 2 minutes for 20 minutes. So then you will see the color change, what happens gradually, and the final observation. Then you can understand how an enzyme acts on a substrate. So students, you have understood the steps of this activity clearly. So let's do this activity in the lab. Now we will start the activity by taking 2 milliliters of the starch solution. So before taking the solution, I will mix it well. Since this is not a quantitative activity, I will take a small amount of starch, about 2 milliliters. To this, we will be adding 2 milliliters of the germinating green gram extract, which contains amylase. So now I have equal volumes of starch solution and the amylase extract. If you all are doing it in a group, one person can mix it. Simultaneously, another student can start the stopwatch, because we will need to time this activity and do the testing every 2 minutes for 20 minutes. So now I will mix solutions and immediately start the stopwatch. So now the reaction will take place between starch and amylase. While we wait for the first 2 minutes to be over, I will show you all the color change that will take place between starch and iodine. Iodine is the reagent that we are going to use for the testing. So this is a starch solution that I used. I take a drop of the starch solution so that you can again look at the color change. To this, I will add a drop of iodine. Now you can see it is a very dark blue color, almost blackish blue. This is only for our reference purpose. Now we will get ready to take the first drop from the mixture and test for the presence of starch. So I have the starch amylase mixture. Now 2 minutes is up. I draw a drop from it. To this, I will add a drop of iodine. Another drop to see the color change clearly. Here also you can see the formation of dark blue color, but comparatively to the starch solution, this is somewhat lighter. A slight difference can be observed. Again, we will have to wait for another 2 minutes to observe the next color change. Now, end of 4 minutes. So, we will take another drop from the reacting mixture. I will place it here. And to this again, we will add iodine. Here also you can see the formation of the dark blue color, but when you compare the 3 samples, the first one pure starch is very dark, after 2 minutes is a little lighter, And you can see after 4 minutes there is a slight difference. We will have to wait for a long time before we see a distinct difference. So until then, we will have to draw samples from the mixture every 2 minutes and test it with iodine. Now we have waited for 20 minutes. Again, we'll test with another drop of the mixture. This is again a mixture of starch and amylase that has been reacting for 20 minutes. To that, we'll add a drop of iodine. You can clearly see that it gives a reddish brown color. So that means from this observation you can conclude that there is no starch in this reaction mixture. So as you know, amylase is an enzyme that acts on starch and hydrolyzes it to maltose. All the starch that was present in the reaction mixture has been now converted to maltose. So that is the reason we do not get a color change. Only the color of iodine is visible. So with this observation, students, we can clearly understand the activity of amylase on starch. Okay, students, now you all observed the activity of amylase on starch. You saw the color change take place when we drew out a drop of the reaction mixture after the first 2 minutes and when we added a drop of iodine solution. You saw that there was the appearance of purple or dark blue color. But with time, when we kept on repeating the steps, the blue color decreased. The purple color or the dark blue color decreased. And finally, after 20 minutes, there was no color change. So that was the observation. So if I write down the observations after the first 2 minutes, dark blue or purple color appears on addition of iodine to the mixture. What happens afterwards? With time, the intensity of the color decreases. Then, with time, the intensity of the color decreases. Then what happened at the end of 20 minutes? After 20 minutes, there was no color change. So after 20 minutes, 20 minutes, there was no color change. How would you explain this activity? Initially, we took starch solution and we added amylase solution. And when we drew out a drop from the reaction mixture and tested with iodine, we got the purple or dark blue color. That means there was starch in the mixture. With time, this intensity decreased. That means the amount of starch present in the reaction mixture decreased with time. And after 20 minutes, there was no blue color. That means there is no starch in the reaction mixture after 20 minutes. The reason is amylase acted on starch and it hydrolyzed the starch to maltose. And maltose does not give a dark blue or purple color with iodine. So the starch that was there initially gave the dark blue color or purple color with iodine, and gradually it was being converted to maltose by amylase. And finally, there was no starch in the reaction mixture because it had been completely hydrolyzed to maltose. So you don't see the appearance of a purple or dark blue color. From this, we can conclude that amylase acts on starch and hydrolyzes it to maltose. So here I will write down the explanation and then we can conclude the observation. So here we can say amylase hydrolyzes starch to maltose. After 20 minutes, there was no starch in the reaction mixture. Because all the starch was hydrolyzed to maltose by amylase. So there we have demonstrated the activity of amylase on starch. So students, now you have understood how enzymes act on substrates. They can hydrolyze the substance to their constituent components, like how amylase hydrolyzes starch to maltose. So here we have discussed the structure of proteins, the significance of proteins, then as a significance, what enzymes are, and the activity of enzymes. So with that, we have come to the end of the discussion on proteins and enzymes. Now we will go on to the next type of biomolecules, that is lipids. Lipids. Fats and oils belong to this group. So this is another type of biomolecule, and under lipids there are two types, the fats and oils. This is based on the way they exist in nature. Lipids which are solid at room temperature are called fats. So if you have a lipid, for example, if we take butter, it is a lipid and it's a solid at room temperature. So butter is a fat. Lipids that are solid at room temperature are called fats. and liquids are called oils. So, if we take any type of oil, coconut oil, gingerly oil, olive oil, all these are oils that are liquids and they are lipids. So, lipids are of two types. The solid lipids are fats and the liquid lipids are oils. If we look at the elemental composition, constituent elements are three elements, that is carbon, hydrogen and oxygen. This is similar to carbohydrates. Out of the four types of biomolecules, both carbohydrates and lipids are made up of only carbon, hydrogen and oxygen. And if you can remember, carbohydrates, the hydrogen is to oxygen ratio is 2:1. But here, the hydrogen is to oxygen ratio is greater than 2:1. So, this is a different ratio. Although it has only carbon, hydrogen and oxygen, the hydrogen is to oxygen ratio is more than 2:1. And all these three elements, carbon, hydrogen and oxygen, combine to form two types of molecules. They are fatty acids and glycerols. And these two molecules or these two substances combine and form a large complex molecule which is the lipid. So, here we can see the combination of carbon, hydrogen and oxygen give rise to fatty acid and glycerol. These two substances combine and form lipid, and when lipid is formed, water is removed. So, here also many fatty acid molecules combine with glycerols, and these fatty acids are also very large molecules. Therefore, the resulting lipid is a very large molecule. And because they are very large and they are made up of these two components, lipids are insoluble in polar solvents including water. If we look at substances that can dissolve other substances, they are known as solvents. They dissolve other substances which are called solutes. And out of these solvents, there are two main types. One is polar. Solvents like water is a polar solvent. Whereas solvents like oil, kerosene are known as organic solvents or non-polar solvents. So, here lipids are insoluble in polar solvents. So, lipid does not dissolve in water. You would have seen if you take a small amount of water and oil into a small bottle and mix it thoroughly, if you leave it at a side for some time, you can see that the water and the lipid or the oil separates out into layers. So, that means lipids do not dissolve in water, but they are soluble in organic solvents. So, as I said, organic solvents can dissolve lipids, both oils and fats. So, this is the structure of lipid molecule, which is again a biomolecule made up of carbon, hydrogen and oxygen. Now, we will look at the food which contain lipids. So, as you know, there are two types of lipids, the fats and oils. So, therefore, there are different types of food. Some contain fat and some contain oil because of their physical nature. So if it's a solid, it will contain fat, and if it's a liquid, it will contain oils. So as examples for fats, we can take coconut, butter, margarine. Then we have the nuts, which are usually rich in fats. For example, groundnut. Groundnut, another name is peanut. That is rich in fats. So we can also include groundnut. And seeds like gingerly also contains fats. Therefore, the oil that is obtained from coconut is coconut oil and gingerly is gingerly oil. So, they are both oils. They are also substances that are rich in lipids. So, we can also include the different types of oils. And other oils also contain lipids. So, these are the facts that you need to know about lipids. Now, we will discuss the significance of lipids. If we look at the significance, it is used as an energy source. So, normally carbohydrates and lipids are the main energy sources of a living organism. Only in the absence of these two, protein will be used as the energy source. But you have to remember that lipids provide more energy than carbohydrates. So, this is one of the energy sources. One of the energy source provides more energy than carbohydrate. So, usually lipids give out more energy than carbohydrate. When we say different structural components, phospholipid and cholesterol are components of the cell membrane. So, to specify, phospholipid and cholesterol are components of the cell membrane. So, as you can see, two different lipids that are present in cell membrane in addition to proteins. Then the next significance is the conservation of water. This is done in different ways. If you have seen, you would have surely seen the leaves, certain leaves which have a shiny nature. For example, aloe vera, cactus, if you look at the leaves, the surface will be very shiny and wax-like in nature. The lipid that is present on the top layer of these leaves are called cutin, and this cutin is a lipid, and this prevents loss of water from the plant. So, therefore, lipids help to conserve water within the plant. So, one significance is conservation of water. Cutin layer on leaves help to conserve water. The cutin layer, you have to remember this name, cutin. that is a lipid that is there as a layer on leaves, and that helps to prevent loss of water. That is conservation of water. Then another significance is the maintaining the body temperature. As we discussed before, there are warm-blooded animals like mammals and birds like us. So we have a fixed body temperature. The body temperature does not change with the change in the environment temperature. So in order to maintain the temperature, loss of heat has to be prevented. So for that, the lipid layer in our body helps to prevent heat loss. There is a layer of lipid, a fat layer under our skin. This is called the hypodermal. Dermis means the skin. So below the skin is hypodermal fat layer. This hypodermal fat layer helps to maintain the body temperature. So this is also a significance of lipid. Then if we look at certain organs like heart, stomach, these organs are covered with a fat layer, and this fat layer absorbs shock and protects the internal body organs. So there is a fat layer that protects internal body organs from shock. So a fat layer surrounding internal organs. It helps to protect the internal organs. Then finally, to synthesize some hormones. We already discussed about hormones under the significance of proteins, and you can remember, students, hormones are substances that regulate or help to regulate homeostasis and coordination. And there are some hormones that are protein hormones, and there are other hormones which are lipid or what we call as steroid hormones. Examples are estrogen, testosterone, and cortisol. So students, these are the hormones that are made up of lipids. So we call them as steroid hormones. So remember, hormones are needed for the coordination of the human body, and also there are two types of hormones. Some are made up of protein and some hormones are lipid hormones. Another name for lipid hormones is steroid hormones. That brings us to the end of the discussion on significance of lipids. Now we will see what are the types of fatty acids present and how they make up the lipid. So as you can see here, fatty acids can be divided into two groups. One is called saturated fatty acid. Saturated fatty acid. When there is a fatty acid or a molecule in which there are only single bonds between two carbon atoms. When you take two carbon atoms which are next to each other in a molecule, they are chemically bonded, and if there is only one bond, that is a single bond, fatty acids which contain only a single bond between carbon atoms is called a saturated fatty acid. But if there are double bonds, when you look at two carbon atoms, there are two bonds like this between two carbon atoms. Then we call it a double bond. These types of fatty acids are called unsaturated fatty acid. So here you can see fatty acids which contain only single bonds within carbon atoms are called saturated fatty acids. And saturated fatty acids exist in room temperature as solids or semi-solids. Whereas the unsaturated fatty acids that contain the double bond, it can be one or several double bonds within carbon atoms, are called unsaturated fatty acids. And unsaturated fatty acids exist in room temperature as liquids. So from this you can understand that usually the saturated fatty acids lead to the formation of fats and unsaturated fatty acids lead to the formation of oils. So this is an extra knowledge that has been given to you, and with this we finish the discussion about the structure and importance of lipids. Now we will see how we can identify or test the presence of lipids in a food substance. So this is the test to identify lipids. Here also we will be doing two types of tests, one for fats and the other for oils. So for this test we use one special reagent that is called the Sudan 3 reagent. Therefore, the test is known as Sudan 3 test. So for this test, we use different materials. One material that we will be using is coconut oil. So coconut oil for oil and butter for fat. We need a test tube to carry out the test. And also we have the Sudan 3 reagent. It comes in form of a powder. We can dissolve it and prepare the Sudan 3 reagent or Sudan 3 solution. Remember students, when you write Sudan 3, we need to use this Roman number 3. We don't use the Arabic number. It has to be the Roman number 3. So we will be doing two types of test. One is for the oil. I will explain that first. What we do is we take a small amount of oil into the test tube. And you can see the oil clearly. It will have a slight light yellowish color. Usually coconut oil has a light yellowish color. And into that you add a few drops of Sudan 3 reagent. That also we add it drop by drop. And when you add each drop, you can see it going down like a fat globule. A globule is a spherical shaped structure. You can see that moving nicely downwards. And then you mix the solution, that is the oil and the Sudan 3 together. When you mix it and look at it, you will see that there will be red fat globules formed. So that will be the observation for the activity to test the presence of oil. I will brief this procedure first. So here we do the test for oils. Test for oil. Under that we take a small amount of oil is taken in a test tube. To this, the second step, students, we have to add the few drops of Sudan 3 reagent is added. So that will be the test for oils. Then you will get the observation. At the same time, we need to do the test for fats. So here we have butter and we will need a blotting paper or a sheet of white paper. So I will include that also here. We will need a sheet of paper. So we can use a normal paper, we can use a filter paper, or we can even use a blotting paper. Any paper to observe the change. So here what we do is, we take the sheet of paper, we rub it on the butter, on the surface of a butter. So a little amount of butter will come onto the sheet of paper. Then we can fold the sheet of paper and scrub it gently. And when you open it out, and if you can hold it to a light, or otherwise if you just look at the paper, you will see a translucent patch, an oily patch. Normally we call it an oily patch. But here that was the result of butter, which is a fat, because we cannot do the same activity with Sudan 3 reagent. We do this second type of activity, rubbing the butter on a sheet of paper, and when you observe, you will get a translucent patch. So that confirms it is a fat substance or a lipid substance. So this is the second activity we will be doing. I will summarize the steps again. So first we looked at the test for oils. Now we will be looking at the test for fat. Test for fat, here we take butter. Here we take a small amount of butter on a sheet of paper and we fold the paper and rub it together. Fold the paper and rub the butter gently. We rub the paper together so that the butter will get rubbed gently, and then we observe the patch. Observe the patch. Patch formed. So this activity contains two parts. One is the Sudan 3 test for the oil, which is a lipid, and the second test for the butter, which is a fat. I hope you have clearly understood the steps. So now we will go to the lab and carry out this activity. Okay students, Now we are going to test for the presence of lipid in biomolecules. So as constituents of biomolecules, we take food samples for our test. And as you know, lipid, there are two forms, the fats and oils. So I am going to use two samples here. One is butter, an example of fat, and coconut oil, which is an example for oils. And the reagent that we are going to use today is Sudan 3. I have already taken a small amount of Sudan 3 into a test tube. You can clearly observe the color of Sudan 3. It is a red color solution. First, a small amount of oil is taken into a test tube. To that, we have to add few drops of Sudan 3. Here you can see the Sudan 3 moving down along the oil, and when we mix it well, you can see the appearance of red fat globules. Here students, you can observe clearly the appearance of red fat globules. I will add one more drop. Here you can clearly observe the red fat globules. So this is the observation for the presence of lipid in food samples. When you add Sudan 3 reagent, there should be appearance of red fat globules as you see here. The globules are going down from the top. So that is a very simple activity to test for the presence of lipid in oil or oil as a lipid substance. Now we will see the test for solid lipids, that is fats. You cannot use the same reagent because this is in the solid state. So what we will be doing is we will use a white paper. Any white paper is okay, and we take a small sample of butter onto this white paper. When you fold the white paper and rub both papers together, you will see this translucent patch. Here you can see a translucent patch. This is the translucent patch. This appearance of the translucent patch confirms that the substance we used is a fat. In other words, it is a type of lipid. Okay students, now you all had a very good look at the observations of the two activities we just did. So we will put that into writing. If you remember, the first observation for the first activity, that was the test for lipids, and that also for test for oils. When we tested for oil, we added few drops of Sudan 3. And now recollect your observation. What did you see? You saw the formation of red fat globules. So when we say test for oil, that is the Sudan 3 test. The observation is formation of red fat globules. Red fat globules. So this confirms the presence of oil in the sample, and this confirms presence of oil in the sample. And then we did the second activity with butter. In that you got the observation. Now try to remember the observation again. There was a translucent patch obtained. So the test with butter you saw a translucent patch. Patch formed on paper. This confirms that it is a fat substance or a lipid substance. And that confirms so under lipids there are fats and oils. We did two activities to confirm the presence of fat and oil in food substances. So with that we come to the end of discussion on lipids. Now you all have a very good understanding about what lipids are, the elements present and their significance, and finally the test done to confirm the presence of lipid. So up to now we have discussed three types of biomolecules: carbohydrates, proteins and lipids. Remember students, there were four types of biomolecules. And what is the fourth one? Yes, it is the nucleic acids. Now we will discuss about the nucleic acids, the structure and the significance.